Right, so we're going to talk about DNA, we're going to talk structure, and then its replication, because at some point in your life, you got bigger. And if all of your cells are supposed to contain a copy of your DNA, then somehow we have to magically make sure that you can copy your DNA into every single one of those newly created cells. So we're going to start with DNA. We will get to that back page of the POGL that you did and talk about why all of those various things are happening. But the only phase that we're going to focus on within that last page of the POGL that you're looking at is the S phase. And we'll come back over this stuff when you listen and talk about the cell cycle video. So we'll get there. First part is just this plain straight structure that you need to be super familiar with in terms of the DNA. So one, if this wasn't already a bullet point that you have memorized, please, please, please put this in your brain that DNA is always going to be found in the nucleus. It's an acronym. It truly stands for deoxyribonucleic acid. And it has those three parts that you did in your bell work question. They are more specified here. I would recommend that you hit the pause button and then you draw this out so you really know what those are. Most of you were able to do that today, but just have that in mind. Down here in the bottom, we're going to start on the bottom here. We have the deoxy and the ribose sugars. Both of these are pentose sugars. So if you know pentose, meaning there are a 5-carbon ring. Yes, there are 6 carbons in here because one is stuck up on the top, but they're really a 5-carbon ring. Take for a moment and look at the deoxy versus the ribose sugar and find the difference. Oh, hey, this is an OH and this is an OH and then this is an OH over here, but this one on the deoxyribose is just an H. Yes, there's a little mistake in this image, but I really like the image, so I just keep using it anyways. This should be an R in ribose sugar, goes for RNA. We'll talk about that guy in our next unit. And then this guy over on this side is deoxyribose, so DNA on this guy. Phosphate group in the middle links one deoxyribose sugar to the next deoxyribose sugar. And then just like you watched in the video for the DNA timeline, or if you haven't watched that yet, please make sure that you watch the DNA timeline video. You will have seen that these nitrogenous bases stick into the middle. So those are the ones that are always in the middle. You either have a group of 2 or you have a single group on the edge of those, and it takes all 3 of those pieces to form a nucleotide. Many nucleotides strung together makes a nucleic acid. One nucleic acid example is DNA. Oop. So then there's all those words. One thing I want you to kind of think about is it a purine to a purine or a purine to a pyrimidine? Which way does it go? So now looking at this one, did you pick up on what column the adenine fell into? What column was the thymine in, the guanine, the cytosine? If you have the slide deck open on a separate slide or you have the notes printed, you can take a peek back at that slide. Is it a purine to a purine, a pyrimidine to a pyrimidine, or one of each? Single rings or double rings? Which one goes with which? It is a single ring with a double ring, and it is always that way. And you can kind of keep that in the back of your mind because it'll be 3 all the way across. So it's 3 circles that go all the way across. As those 3 circles go all the way across, it gives enough stability for them to stick together to one another, and then for that ladder to twist upon itself. If they weren't stuck together in the way that they were, the double twist wouldn't be able to happen inside of there. So they are stuck together that way. And they are always stuck together in this order for this reason, and it has to do with chemistry, which you don't need to understand. You just need to know that it is always an adenine with a thiamine because an adenine has 2 available spaces to bond to something. Thiamine has 2 available spaces to bond to something, so those 2 stick together. Guanine has 3 available spaces to bond, and cytosine has also 3 available spaces to bond. The thing that is available to bond with one side to the other is always a hydrogen. So it is those 2 that will stick together, and it is hydrogen bonds that form the stepwise ladder in the middle of the DNA strand. I also remember it because adenine and thiamine put together spell a word, A-T, at. How many letters long is the word at? 2. How many bonds are there? 2. That's my shortcut way. Take it or leave it. So, quick quiz for you. This should be very familiar because it is that last table that you filled out on the Poggle, but hit the pause button and you should be able to do this in your head. And what you should have reasoned is the same thing that you did on the back page of that Poggle, but just a quick check for you again. Everywhere there's an adenine, you're going to find a thiamine. Therefore, we should have 30% of our sample being made out of thiamine. 30% of adenine, 30% of thiamine means that is a total of 60%, leaving 40% behind to be divvied up between guanine and cytosine bonded together. So 20% should have been the number that popped into your brain straight away. It's okay if it didn't. You can erase that table, provided you didn't do it in pen on the back page, and you can just practice, try that one again. But the principle is pretty simple on there. Bonus quiz from the DNA timeline video, if you have watched that, is whose rule is that? Who figured out that table in there? Oh, what's that guy's name? He was the last one in the video that we paused for. This guy, Erwin Schargraf, and that was 1947. So A with T, C with G, if we give it his rule, it's just that the percentage of each base equals the percentage of its complement. Remember that the twisted ladder was discovered, thank you, Miss Franklin. Gave us that beautiful photo 51 showing us that the rungs of the ladder were on the inside. Those nitrogen bases had to be on the inside. And as that circle swirled around, it also only had enough space for there to be 2 swirls of the DNA. Technically, on those 2 swirls, there is a major groove and a minor groove. So there are, as it twists around each other, every swirl alternates between a close together swirl and a little bit further apart swirl and a little bit closer together and a little bit farther apart swirl. We also know the exact distance between every single rung on that ladder. It is 3.4 angstroms, so it's a very, very small measurement. It is smaller than a picometer, so it's absolutely itty bitty. We literally had to make up a new unit because it's such a small measurement of distance, but it's also an incredibly important measurement of distance on there. All right, another question that was on your packet, and it was B, it's on the back of the second page, talks about, are these pieces as they fit together, are they parallel or are they anti-parallel? And the answer to that question is they are anti-parallel. And you should have noticed, if you look at the model on the front, that some of those pentose sugars were upright and some of those pentose sugars were upside down. So if you really think about it, put your hands out in front of you and turn one of them over, and your thumbs really lock together and they fit within there. That is what is known as anti-parallel. How we figure that out is off of that pentose sugar. We actually number all of it. So the part where the base pops off of it is called the first prime carbon. And we go all the way around, and then that carbon that pops off of the other end of it that I talked about in the very beginning, that is the 5th pentose sugar, 5th pentose carbon, and then there's a 3rd pentose carbon. So between those two, if the 5 is on the top or if the 3 is on the top, that's how we determine that one side is the parallel strand and then one side is the anti-parallel strand. So there's some naming conventions that are coming here in a second, but that's where the parallel, the anti-parallel come from. That's how we number all of those guys as they go through everything. As they hook together, so anti-parallel means it's symmetrical. It does look the exact same on both sides. One side is just right side up and the other side is upside down. So just that's a 180-degree turn if that helps it stick in your brain a little better. With that directionality that is on there, we have done these pieces. There is something called the read direction or the read frame. You might have heard it called. I think of it as the read direction. And it is that you go from the 5' to the 3' when you are reading down the strand of DNA. That's the side that matters. That's the side that we're going to read, that's going to code for your traits. That'll be really important in our next couple units. We're going to read 3' to 5'. Okay. For DNA storage, in terms of that location, one little extra bonus piece that is in there is keeping in mind that for prokaryotic organisms, remember, they don't have that nucleus, so the DNA is just kind of hanging out there. That's a picture of what it actually looks like in there. In eukaryotes, we not only put it inside the nucleus, but we wind that bad boy up very, very, very tightly, very organized fashion on that guy. You technically have 3.2 billion base pairs in every single one of your cells. You do not use all 3.2 billion base pairings in every single one of your cells. You only use the portion that is needed based on what cell type it is. Regardless of that, you have all of the genetic information in every single one of your cells. So to keep everything hyper heckin' organized, your DNA is broken into chromosome pieces. And some of those chromosomes are loosey-goosey, so we can access the information, and some of them are very tightly bound. Regardless of the loosey-goosey or the tightly boundness to them, this is what happens. This orange dot here, there's actually 8 orange dots in there. Those are called histones. Histones are proteins. You complex 8 of those histones together, and we call that a nucleosome. That's the word on the very bottom here. That's what the DNA wraps itself around so that everything stays really neat and organized. Your DNA is not like your unwound pair of headphones or your unwound computer charger that you can just jam into your backpack. It's real fragile. It's real special. It needs to be treated nicely and with respect, because if you damage that stuff, not good. We're talking things like cancer or other diseases and disorders that might happen. So keeping things really nicely, neatly wound up is really, really important. And that's the structure around which things wind. Your textbook has a much larger, very beautiful picture of this process and what exactly happens. So I would crack open your textbook if you have it nearby and take a peek at, I think it's like a 1.5-page spread that shows everything in there. All right, one other little bit is that sometimes don't forget that DNA can also exist in those little plasmid forms. Remember, we find it inside of chloroplast if you are a plant, and mitochondria if you're a plant, and then mitochondria if you are an animal organelle. Bacteria also have those plasmid pieces, and you can see the little ones designated right there with the arrows that are off of there. So don't forget about those guys. A cool, fun fact with your chromosomes has to do with, we'll talk about that whole thing about why you get shorter when you get older. Because what's up with that guy? Well, the deal is every single one of your chromosomes, because it's super important, it's super fragile, actually has, and you can see it in the picture here, these black regions on the end of them. And it's just this big long series of A's on the end that are there. It's just a ton of A, like literally just adenines, like it's like 200 adenines long that are on the end of those guys. Every time that you replicate your DNA, like we're going to talk about here in just a second, you lose some of those A's. And then you lose some more of those A's, and you lose some more of those A's. And you can see that in this picture here that, yes, I know it's from the Wall Street Journal, but it's still, this is a really good illustration of what happens. And it's just off the end. That little region on the end is called your telomere. You can see that right here in the diagram a couple places. Those telomeres just lose a little bit of their A's every time they replicate. Better to lose a little bit of your telomere than it is to lose any of your genes. But as you lose those, you do end up getting a little bit shorter. So that's not so great. That's kind of a thing when people are looking at like, ooh, what's the magic elixir for life? Or how can I stay youthful forever? It's the whole idea of like not losing that telomeric region off the end of your chromosomes. You theoretically could become ageless if you didn't have to lose any of those parts. Nobody's actually figured that out yet. It's just kind of an idea, a theory that is in there. So a little bit more about telomeres here, if you find that those are really interesting, and definitely an area of research that you could get into, as they are pretty fantastic. Potential spot for also some cancer prevention might be in there, or a way to treat cancer if you do end up with it is by prematurely aging that chromosome and like getting things to get off of it. I'm not sure. Whole big area of research. All right, done with structure, on to replication. So hopefully you remember chromosomal replication from 9th grade biology, where we may have grossly oversimplified the process. We gave you the basics, but just FYI, we did not tell you the truth. So here's the truth about how replication actually happens. 1, also should be a little thing from your memory bank, should be in the S phase of when everything happens. Remember that we need to make bigger people, bigger plants, bigger deer, bigger bunnies, you name it. We need bigger organisms. Organisms, multicellular eukaryotic organisms, as they get bigger, need more cells. Well, one of the things that you need in every single one of your cells is another set of instructions. So we're going to have to replicate those instructions. Things will happen in your life, you've probably already experienced this, where there's a boo-boo or a wound that happens, and you do not, I am going to assume right now, have any of the open, gashing, bloody cuts, scrapes, wounds, you name it, that you gave yourself when you were 2, 6, 10. And that is thanks to a really cool process that we'll study here in just a little bit. One of the pieces of that process is that you made another copy of your DNA so that the cell, that wound healing that happened for you, could take place in all of its perfect glory and that you would not be left with any scars. Because I bet there are major spots where you have scraped yourself and you don't have any memory of it and you don't have any visible sign. And that is thanks to this process that came through and happened and sealed everything back up. The third piece is that you sometimes pass on your genetic information. It was given to you, and you are going to pass that on as well. And we do that when we make babies, which none of you will be doing anytime soon. Thank you very much. It's got a special name for that process. We'll get to that in a little bit. It's a pretty intricate, really cool process, but if you're going to have a baby, you're going to need to give it some DNA. So there's another reason that we need to replicate our DNA. When we are doing all of that stuff, and yes, everything in your cell replicates. We'll talk about that later. It is only happening right here in this tan wedge of the circle called the S phase. If you are going, oh, S phase, I just wish I knew what that stood for. It stands for synthesis. So to make, to synthesize something means to make it. So we are synthesizing new sets of your DNA. So that's when it happens. DNA, as it replicates for you, replicates it using this model over here. So it starts off, we have a beautiful blue piece of DNA, and then we're going to rip that blue piece of DNA, and we are, and you can see, winding into yellow pieces of this. It is known as the semi-conservative model for replication. So we're going to take half of your DNA, and it is going to be an original copy. We're then going to pair that original copy, since we have those base pairing rules. Thank you, Erwin. That all A's must pair with T's and all C's must pair with G's and vice versa. Therefore, all I really need is half of the strand, and then I can make a copy on the other side. So semi-conservative replication is how your DNA goes through and does that. It did take us a while to figure this out. You should, at this point, hopefully have watched the other video. If you haven't, you will soon. And the video that you're going to watch are the two gentlemen that figured this out, because somebody had to have an idea of like, hey, how does that work? Well, Mendelssohn and Stahl are the two gentlemen that had this idea, and they went through and they did all the testing for, well, how does that replicate? Is it just one old strand and then it just photocopies itself? Does it open itself? Does it chop itself into pieces? In the video, they're going to talk about the experiments that they did to figure this out and the ways that they, again, proposed ideas that were wrong. Just like with figuring out the structure for DNA, we do a lot of things that are wrong. You got to throw out a lot of bad ideas before you're going to find the one that's the correct one. So we laugh at all those ideas of like, haha, nitrogenous bases on the outside or a photocopy machine inside of your cells. That's preposterous. Yeah, but you don't know what you don't know. So now we think it's so simple, but somebody had to go through and figure it out. So Mendelssohn and Stahl are who we have to thank for figuring out that it is this blue one in the middle, the semi-conservative model for replication. And then this is their video. If you have not watched it, I would maybe hit the pause button and go open this and listen to both of these gentlemen. I have not done my homework to see if those two are still alive yet, but they are of the same era as Watson and Crick. So they are right on the edge of being alive versus not alive. We're going to move this one along. All right. I love this cute little intro to this. I'll let you just watch it for a second here. What's happening? What do you notice is the same? Who are the people? What are they doing? What's different? And then there was a little name on the end. We're going to come to that guy on there. All right. These are those vocabulary words that you need to know. So we have a thing called a leading strand, and then we have a thing called a lagging strand. And then another brand new term that you need to know, which is called Okazaki fragments. So we'll get to all of these guys. Leading strand is going to be the 5 to 3 direction. It's the same as the read strand. We'll get to read later. Just know that leading is 5 to 3, and it is this continuous synthesis of your DNA. Lagging is going to go the other direction. We'll talk about the lagging because it's going to be this like funky, it literally lags behind is how you can kind of remember that. You need to stick this in your brain that leading is 5' to 3' and lagging is 3' to 5'. You should probably stick that on a flashcard, and then you should probably start quizzing yourself today. What is the difference between those pieces? Okazaki fragments are a name that we give to the fragments that come off of the lagging strand. We'll talk about those here in just a moment. So a couple locations to orient yourself. Yes, we are 100% in the nucleus, but we are not just in the nucleus. Inside of your nucleus, if you did not know this, there is a special place called the nucleolus. And the nucleolus is where we synthesize new nucleotides, both DNA and RNA. There is a nuclear envelope, so all of your organelles are nicely bound up. The nuclear envelope is the one that we call the outside of the nucleus. I know, crazy name. You're welcome that one was easy. And then don't forget that there are pores, so just little holes on the outside of those nucleuses because we have to get the information to get out of your DNA at some point. Not today, but I just want you to have all those orientation pieces on the nucleus. Getting things in and out of the nuclear pore comes after spring break. So here's how it works. We are going to take a helicase enzyme. Hopefully that name, you're like, hey, that was the same name from the beginning little thing. Yes, it is. And we are going to unzip your genes. And this does happen multiple times over, all over your DNA strand. You can see this in this actual picture here that is of a slice of DNA. You can also see it in kind of the more linear cartoon image that is on there. As things are unwinding, we're going to unwind from the AT regions. Why there, you ask, I hope. Wait, well, what's true about A's and T's and the bond that's between those two? Oh, hopefully what popped in your head is like, oh, A to T is only 2 bonds. It's literally more energy favorable for the cell and for the DNA to go ahead and split those 2 bonds as opposed to having to split 3 hydrogen bonds. I mean, it's not like Superman here, but it does take a little bit of effort on the part of the cell. A little ATP has to be spent to split those couple of bonds. And again, we can do this in multiple places all over your DNA because we got to get this process moving here. And you have 3.2 billion base pairs. We got to unwind them off the histones. We got to get the whole thing replicated. We're going to need multiple helicase molecules so that we can get in and do all those pieces. So helicase actually looks like this. It kind of slides along the DNA and is able to melt that DNA open. Then replication fork is on there. Notice there are also now in this third image green dots that are all over there. Those green dots are called single strand binding proteins, and they are just little teeny tiny proteins whose whole sole purpose it is to keep the strands apart from each other because we're trying to make new copies of things. And I don't know about you guys, but if you're trying to read the textbook and then your, cat, mom, sister, annoying brother comes by and they keep closing the book, it's really hard to read and really hard to figure out what you need. So what they do is there are these little proteins that just stabilize that open after the helicase has come through. And so they're called single strand binding proteins. Then what's going to come through, if you've ever painted a house, you'll know this. You know that you have to prime before you can paint. So your DNA also has to get primed before it can get copied. It just needs a little signal that says this is where to start. So it's not so much that there's anything getting laid down on there. It's more of just like a flag of like, go here. And so it's kind of a bad analogy to use the paint, but it's the one that I got because primer is, we typically think of primer in painting, but it's more like a flag in the ground that says, hey, start here. And so you can see the whole thing in this picture. There's something called topoisomerase. Don't worry about that guy. Helicase is coming along. You've got the single strand of binding proteins in there. And then there's this enzyme, primase. Primase is going to hop onto there. I like this image on the other side because it just shows you how close everything is together. This stuff is not far apart in distance. It's very, very, very close. Down here in this picture, it's giving you how many bases away from it each other everything is. So 1 to 63, and then 63 to 271, that's all your primase. So it's very, very close together. Remember, all this stuff is it's itty bitty, teeny tiny. Once primase puts that flag in the ground, then this other enzyme called DNA polymerase can come, and it actually like latches, it kind of hops on. You can see what that molecule looks like. And it is so amazing. It is super cool. It is a triple function enzyme in here. It will both remove the primer, synthesize new DNA, and then edit itself to make sure that it doesn't make any mistakes. So it's doing this unbelievable, amazing job going through and just making sure that absolutely everything is proper and newly constructed so that you have 2 new pieces of DNA used later for a variety of things that we'll talk about. So those guys are doing a lot of work here, polymerase. So polymerase is adding the nucleotides, it's proofreading itself. If it finds any T's bonded to a C or a G, it takes care of those. First go around, it makes 1 in 10,000 mistakes. So not a lot, but like also kind of a lot if you think about the fact that you really can't handle any errors and you have 3.2 billion base pairs and every 10,000 gets a mistake. That's going to add up. So we don't like that. So thank goodness it comes with its own proofreader, kind of like you're your own proofreader before you turn in an essay, I hope. And so it takes that down to like 1 in a billion. So we went from 1 in 10,000 to 1 in a billion. It really does a very good job of editing itself and making sure that there are no mistakes that are in there. I mean, it's not perfect, but it's okay. 1 base can be a pretty big deal. So sickle cell anemia, we're going to study this later, is a single base pair change. It just took 1 mistake, and we now have this worldwide disease called sickle cell anemia. We're going to talk a whole lot about that in our next unit. So if you have sickle cell anemia questions, save them or go look up sickle cell anemia and know that that's what we're going to do after spring break. All right, so leading versus lagging. Okay, we are trying to go 5 to 3. We love going 5 to 3. That's the easier way to go. The reason is that is when primase latches on, it can actually smoothly move in that 3' direction. The end of that carbon strand is smooth for it. When we go in the opposite direction and we have to read from 3' to 5', that 5' end has a structure on the end of it. And so it gets stuck on that polymerase. So what actually happens is you go in these, I like this image the best, you have to move over and then you synthesize backwards, and then you go forward and then you synthesize backwards, and then you go forward and then you synthesize backwards. The person who figured that out, his name is Okazaki. He's a Japanese scientist, and he figured out this difference between the leading strand going smoothly and the lagging strand getting chunked in terms of its replication, or what we call discontinuous synthesis. I just think of it like chunky. Just you just have to do it in a slightly different fashion. So these are just two different pictures to help you. Your book also has a fantastic picture and explanation on those Okazaki fragments and how he figured all those things out. Technically, there's some differences between the DNA polymerase that's on each one of the strands, but don't worry about it. The last thing that happens is that RNA primer that hops on there that tells DNA polymerase, like, come here. We have to get it out of the way, so we do, and then we have to seal all of those discontinuously made fragments with ligase. What is something that you seal things up and together with? If you just thought of glue or a sewing stitch or tape, that's what I like in ligase too. It's just some sort of seal, something that you put down that smooths those discontinuously synthesized fragments together. So like this picture, because it just kind of gives you that big highlight on the two different ways that those things are made. And then don't forget that there's those telomeric regions that always end up on the end of both of those. When you get to the very end and you are done, we got to hop off and we got to finish everything up, and we got to like give us our thumbs up. So we take those primers out, the ligase fills in there, and we now have two identical copies of your DNA made off of what is known as a template strand. So this is what it looks like at the very end when you're done. Also, again, just a little shout out to Mendelssohn and Stahl for the work that they did figuring out this process, because kind of like DNA, this one was heckin' complicated and really only had the two of them just doggedly working to figure this out. DNA, yeah, it took 7 people, but these two, just 2 people that we give this credit to. So that is on there. Just to give you the real picture of like what it actually. I think we have enough in because we have like 3 So even they didn't tell you the whole truth. If you think it's fascinating, I think that it is absolutely fascinating. In grad school, we did learn a whole host of other enzymes that really get everything to work and everything that's going in there. There is a lot more that you can do in there that's really amazing with it. So I would ask yourself this practice question. See if you can figure out the answer to that guy. And then I would play match the enzymes. You should know what every single one of these enzymes does, where is its function, how does it enable the cell to do a better job of its replication time, like how does it make things work in here. When you are done practicing with both of those guys, and all the answers are in the notes, so open up the PowerPoint separately and then go through and do those. The formative is open for you. Let me know if you have questions on that formative. Ask each other, work together, and I will see you soon.